Good morning, everyone. I would like to welcome you to the fourth webinar of the Neuroimaging Webinar series, jointly organized by NeurATRIS and NeurATRIS. Today, we are very pleased to have two lecturers from NeurATRIS and Celphedia speaking about in vivo preclinical models for the study of neurodegenerative diseases. I would like to make you aware that this webinar is being recorded, including the questions and answers at the end of the session. It will be made available on NeurATRIS and NeurATRIS websites shortly after this session. We have reserved some time at the end of this session for questions. We invite you to write down your questions in the question box in the control panel as you see on this slide. Before I introduce our speakers, I will briefly present the three research infrastructures bringing you this webinar today. NeurATRIS is the European infrastructure for translational medicine. NeurATRIS helps you de-risk and add value to your drug, vaccine, or diagnostic development program. We do this by providing fast, tailored access to cutting-edge enabling technologies in translational research. Via our central hub, you can access the vast array of clinical expertise and high-end facilities that are available within the more than 80 top-tier academic centers across Europe. Always keeping the patient at the heart of our interests, NeurATRIS relies on three pillars to help early decision-making: biological insight, validated analytical tools, and clinical observation. NeurATRIS' strategic research agenda follows two main directions. From the technical perspective, we help select and validate next-generation translational tools and biology. From the operational perspective, we foster new collaboration and encourage new funding models. You can find out a lot more on our website, neurATRIS.eu. NeurATRIS is the French node of the European infrastructure NeurATRIS, and it is the only node whose specialization is solely neuroscience. NeurATRIS is also a national research infrastructure dedicated to neuroscience, and more precisely the neurodegenerative diseases that are Alzheimer's, Parkinson's, Huntington's diseases, and multiple sclerosis. NeurATRIS is composed of expert teams from 5 renowned hospitals and research centers, accessible through a one-stop shop. This consortium brings together all the needed bricks to build a full translational neuroscience project and gives you an easy access to multidisciplinary expertise and high-level platforms to achieve your project in neuroscience from bench to bedside. All this information and more can be found on our website. Should you have any question about NeurATRIS, please feel free to contact me or our business development manager, Ali. We will be happy to answer them or to put you in contact with the experts you need to define your project. Celphedia is a French national research infrastructure distributed throughout France, but also a unique portal giving access to the best model among 3 big families of model organisms: non-mammalians, including aquatic vertebrates such as zebrafish, rodents with mouse and rats, and non-human primates. The aim is to help researchers to choose the best animal model to answer their scientific question. To do this, massively innovative and standardized approaches have been developed to understand the human genome, to create human disease models, and to promote therapeutic innovation. With 15 distributed nodes, Celphedia gathers a combination of expertise and skills in these domains: creation, meaning generation of genetically modified model organisms, husbandry and breeding, phenotyping, including functional exploration, archiving, distribution, and adapted trainings, including regulatory trainings for the use of animals on a scientific purpose. Celphedia offers a large catalog of cutting-edge services nearby the users with standardized processes. It is open to the whole scientific community, supporting many areas such as basic research, biomedical, preclinical proof of concepts, all this while complying to the highest standards of welfare and ethics. Our first speaker today will be Dr. Marie-Christine Birling. Dr. Marie-Christine Birling leads the genetic engineering team at the Institut Clinique de la Souris in Strasbourg since 2003. She advises scientists on the best approach for generating relevant rodent models. She has initiated more than 1,500 à la carte models, leading to a number of high-impact scientific papers. Since 2013, she has implemented the CRISPR-Cas9 technology in her lab, and most of the models are now generated through this approach. Our second speaker will be Dr. Romina Aron Badin. Dr. Romina Aron Badin directs both the primate neurosurgery and behavioral platforms in MIRCen in Fontenay-Rose near Paris since 2008. She has been PI of over 30 projects involving non-human primates and has extensive technical expertise in experimental design and execution of preclinical protocols for brain delivery of drugs, viral vectors and cells, as well as in the assessment of cognitive and motor functions in primates. Her current research interests include functional evaluation of innovative gene or cell therapies for Parkinson's and Huntington's diseases, as well as the development of new primate models of Alzheimer's and Parkinson's diseases. I will now hand over the presentation to Marie-Christine.
Hello, everybody. In this talk, I will speak mainly about neurodegenerative disease, but also I want to explain you why I believe mice are still good models to study different human diseases. Ongoing studies in many species seek to understand the origins and consequences of phenotypic variation under normal and dysfunctional conditions, with the aim of identifying targets that can prevent, stabilize, and reverse disease. Some suggest that only humans are appropriate for studying these questions and argue that candidate drug targets identified in mouse models are largely unreliable. In this webinar, I will show evidence that mouse models continue to make fundamental contribution to our understanding of genetic principles, pathogenetic mechanisms, and therapeutic modalities. There is a virtuous circle in which the power of observational studies and natural experiments in humans are closely integrated with the rigor and true experiments in model organisms. There are challenges in the human genetic. There is a limitation in the availability of biological material. We cannot get any material from unaffected individuals because when humans have disease, we cannot take samples from them before the disease. Cells and deep tissues are inaccessible and there cannot be time point before the pathology. Time course studies are logistically challenging. Large sample sizes are needed and this introduces genetic and environmental heterogeneity. Observation can be variable between individuals. And finally, in vitro studies are impossible because critical cell types are not known. Indeed, combination of cells or communication between tissues and organisms is difficult to arrange in vitro. The challenges in human genetics. Models of complex system, big data, need to be tested experimentally in order to evaluate assumptions, predictions, and hypotheses. Model organisms allow to conduct the experiments needed to validate human discovery. Laboratory animals allow a level of experimental control not possible in humans. For instance, in the case of the mouse genome is well characterized. The mouse genome can easily be engineered, and it was the only one that was possible to engineer for ages. Genotyping of the mouse is easy, and there are plenty of phenotyping toolkits. You can go on our website PHENOMIN, but also on the International Mouse Phenotyping Consortium website, in which you will find many information, and I will discuss that in the next slides. Why mouse models? As I said, for decades, the mouse was the only mammal in which targeted genetic modification were possible. Indeed, in the case of the mouse, we had ES cells, embryonic stem cells available in specific genetic background, and it was so possible to precisely modify, alter, correct a gene through homologous recombination and select a rare event in ES cells, and after, of course, generate a mouse line. That was not possible in any other species. Since the early years 2000, of course, genome editing, different nucleases, including CRISPR-Cas9 technology, arose. And so now targeted genome editing is possible in a wide variety of genome. And that's not only mouse, but it's rat, donkey, sheep, monkey, and plenty of other species, and even human. The only limit of the use of the nucleases is ethic and what is allowed by the ethic use. I think we need to better educate the population to explain the benefit of studying different species with the use of these genome editing tools. First, I want to show you an example of what we can do in mouse that is difficultly possible in other species, and I will show you one example in which we visualize the expression of a gene. In this case, it is the characterization of the GPR8 Venus knock-in mouse line, a line that was done at PHENOMIN-ICS. GPR88 is an orphan J protein couple receptor originally characterized as a striatal enriched transcript and is a potential target for neuropsychiatric disorders. The Venus cDNA was fused directly before the stop codon in exon 2 in order to generate a fusion. The Venus fusion does not alter GPR88 mediated J protein activation as shown on these binding experiments, whereas it is clearly affected in the case of the knockout model. In brain sections from Venus GPR88 Venus homozygous animals stained with an anti-Venus antibody, the GPR88 expression is detected in the olfactory tubercle, in the nucleus accumbens, in the cortex, and in the caudate putamen. GPR88 has a prominent role in multisensory processing. Because sensory integration is disrupted in many psychiatric diseases, this study definitely positions GPR88 as a target to treat mental disorder, perhaps with activity on cortical sensory networks. Now, I want to explain you a little bit what the IMPC is at PHENOMIN-ICS and see if we are part of the IMPC. Although next generation sequencing has revolutionized the ability to associate variants with human diseases, diagnostic rate and developmental of new therapy is still limited by a lack of knowledge of the function of most genes. The IMPC, International Mouse Phenotyping Consortium, is creating a genome and a phenome-wide catalog of gene function by characterizing new knockout mouse strain through a broad set of phenotyping tests. You must know that all mice generated in the IMPC are available to the biomedical community. Analyzing 3,300 genes identified model for 360 diseases, including the first model for type C Bernard-Soulier syndrome. 90% of the phenotype annotation were novel, providing functional evidence for more than 1,000 genes and candidates in genetically uncharacterized disease. 135 strains had phenotype overlap with human disease with unknown mechanism, and therefore are novel Mendelian disease candidates. Out of more than 2,500 genes with non-lethal IMPC phenotype, IMPC data provides new functional knowledge for more than 1,000 of these genes. Then it's really important, this IMPC, and I really advise you to go on this website. You will find plenty of nice information. The work of the IMPC highlights the continuing importance of mouse models in understanding disease mechanism. Mice are vertebrate mammals with physiological characteristics that recapitulate those of all major human biological systems. They allow the study of processes that cannot be investigated in vitro, including the behavioral, inflammatory, endocrine, and sex-specific processes on disease. Inbred mouse strains such as C57 Black 6N strain used by the IMPC have standardized uniform genetic backgrounds that decrease phenotypic variability. And that is something we cannot see in human because we are all very different and we have plenty of SNPs that makes us different. Then working in an inbred background renders the analysis much easier and the size of the cohort much lower. Most strains have a 2-year lifespan that allows comprehensive studies in a timely manner. A substantial fraction, something like over 15% of the mutant strain, will be re-phenotyped in mice at 12-18 months of age to identify genes involved in late-onset disease. We sometimes forget to phenotype animals when they are older, and I think it's really important to go and see what a gene defect does in later ages of the development. Another very interesting paper published in 2016 by Dickinson and collaborator describes the new developmental phenotypes. On over 1,700 unique gene knockouts, 410 were found lethal. Lethality occurred at different stages of the development. You can see it here. Analysis reveals that incomplete penetrance and variable expressivity are common even in a defined genetic background like B6N. And that's the case here. That's this mouse that we call subviable. This means that some of the animals are dying when they are homozygous, but some are also surviving. For all cases of lethal and subviable genes, full cohorts of heterozygous mice will be phenotyped as part of the MPC adult phenotyping pipeline, along with surviving subviable homozygous. You can see them here. In some cases, viable homozygous mice display a greater number of phenotypes hits per gene than heterozygous mice from the lethal class, although the average difference was only of 1.4 more hits. There is a virtuous circle which was well discussed in a review of Nadeau and Howarth in 2019. This virtuous circle relies on forward genetic, which starts from phenotype to discovering of genes, and reverse genetics, which starts from gene and goes to the discovery of phenotypes. Discovery in humans and in models organisms triggers a recurring circle of observation in human, experimentation in mice, and finally validation in human. In some cases, true experiments are possible in human, but in most cases, these experiments are not feasible in model organisms such as mice, where properly controlled study designs with adequate statistical power can be arranged. Here, for example, I show you some disease gene discovered first in human. And that is the case, for instance, CFTR, which was the cause of cystic fibrosis, or for instance of Huntington, which gives Huntington disease, but also example of gene studies first in mouse that were later shown to be relevant to human disease, like FTO, that leads to obesity, or KIT ligands that leads to testicular cancer. The contribution of mouse models is important in neurobiology and neurodegeneration. Discoveries that phenotypes are often reversible have demonstrated the feasibility of intervention in humans. Here is an example of a disease. Infantile spasms are a catastrophic form of pediatric epilepsy with inadequate treatment. In patients, mutation of ARX, a transcription factor selectively expressed in neuronal precursors and adult inhibitory interneurons, impairs cells migration and causes a major X-linked infantile spasm syndrome. Using an animal model, the ARX GCG 10+7 mouse, Olivetti and collaborators determined that a brief administration of estradiol during early postnatal development prevents spasm in infancy and seizure in adults. Estradiol was effective when delivered after puberty or 3 days after birth. They concluded that postnatal estradiol treatment could potentially serve as a therapy for inherited interneuronopathies. That's really also a nice example of what mice can bring to human disease. Many neurobehavioral disease genes have been subsequently validated in humans. Here is another illustration with MPTP2. MPTP2 duplication syndrome is one of the most common genomic rearrangement in males and is characterized by autism, intellectual disability, and motor dysfunction, anxiety, epilepsy, recurrent respiratory tract infection, and early death. The correction of MPTP2 in mouse model largely reverses the behavioral, molecular, electrophysiological deficit. Okay, and here I will show you how it works. A transgenic mouse was generated with the overexpression of human MPTP2. And this mouse, here it's a case of a male MPTP2 is an X-linked chromosome. And this mouse was bred with a fluxed MPTP2 and also bred with a ubiquitous CRE-ERT2, inducible CRE-ERT2. The males were analyzed, and you have here one copy, which is normal. In this case, it is flux, and you got animals with a transgene plus one flux copy, and you got animals with a transgene plus one flux copy, which is two copies that is like the disease. And when it's bred with tamoxifen, because there is a CRE-ERT2, you go back to one copy. And you can clearly see here on the Western blot that having effect of the CRE, it deletes one copy of MPTP2 and it goes back to a single copy and it goes back to normal. The level of expression is almost normal. You can also see that on this immunohistochemistry, you can see that the control, which is the fluxed animal with tamoxifen but with no CRE-ERT2, it's like the wild type. And when we use the animals that has a transgene plus the flux allele plus the CRE-ERT2 with tamoxifen, that the level are approximately the same. And that was also shown when the behavioral tests were done. And when you look at the open field, you can clearly see that the almost wild type animals that are the conditional animals behave almost the same like the animals in which we had the duplication, but we went back to one copy with the CRE-ERT2 and the tamoxifen. Then I thought that was a nice illustration that I could show you. Another nice illustration here is DIRC1A. You must know that individuals with partial human chromosome 21 trisomy and mouse with partial chromosome 16 trisomy, you must know that chromosome 16 trisomy is partially synthetic to chromosome 21. And these trisomies contain an extra copy of DIRC1A gene present in various alterations of brain morphology. They present also learning impairment modeling of Down syndrome. A model of overexpression, human YAC transgene, which contain human DIRC1A plus 5 other genes, shows that DIRC1A is involved during development in the control of brain volume and cell density of specific brain region. Quedge and collaborators demonstrated that chronic administration of polyphenol from green tea can have a corrective effect on brain alteration. The diet is bringing the level of active DIRC1A to a value that is in between the value produced in transgenic and in the wild type animal. Okay? It's almost the same. That's really interesting. After, when they did object recognition test, in this test you put each mice in an open field setting in which one object is placed at the left-hand side and a second object at the right-hand side. The test comprises 5 daily sessions first, and after the training session, an object is substituted to a novel object. Wild-type animals showed a clear long-term memory acquisition during the retention period. Here. By contrast, the memory index was close to 0 with water-fed transgenic animals that had 3 copies of DIRC1A. These animals were unable to recognize object novelty in the object recognition task. It's here. The GTP-treated transgenic mice had a memory index equivalent to that of wild-type mice. Then, when you administer some green tea leaf, you can see that it goes back to almost normal. Then, the polyphenol molecule is called epigallocatechin galate, EGCC, and it was purified from green tea and was found to be a specific and safe DIRC1A inhibitor. Then here is a study, always on DIRC1A, published in Lancet, that shows that the compound we just discussed about, EGCG, reduces DIRC1A overexpression for people with Down syndrome and achieves an important cognitive function. Adult aged between 16 and 34 years with any of the Down syndrome genetic variation, trisomy, partial trisomy, mosaics, or translocation, were tested in double-blind, placebo-controlled phase 2 trials. Participants were randomly assigned to receive EGCG, 2 mg per kilo per day, or placebo. All patients received a cognitive treatment for 12 months. At 12 months, participants treated with ECGT and that had cognitive training had significantly higher score in visual recognition memory, inhibitory control, and adaptive behavior. No difference was noted in adverse effects between the treatment groups. Phase 3 trial with a larger population of individuals with Down syndrome will be needed to assess and confirm the long-term efficacy, EGCG, and cognitive training. And finally, to go even a little further and perhaps to be a little controversial, what occurred is that some babies were given green leaf supplement, and they were shown to be likely to develop less strong facial feature of Down syndrome. And then antioxidant EGCG made 6 out of the 7 patients with healthy facial feature. Then they look less like having a Down syndrome face. Only when given before 3 years old, due to the face development still developing before 3 years, they showed this kind of effect. Of course, afterwards, it didn't work anymore. Of course, this is controversial because it's a kind of normalizing of the patient, and some parents don't like so much the idea of doing that. But anyway, no, I'm finishing my talk. I'm just saying it is also, I think, I hope I showed you that mouse is still really important. I have plenty of other examples, but I want to emphasize here that it is also necessary to work on more complex animal model or in human. It is, of course, even more complex to work with more complicated models like monkey, and there are a lot of ethic concerns and discussion. But you must know, and that's because my main work is to do genetic engineering and genome editing, that no, it is possible to modify any animal models with CRISPR. And I think it's just my believing that it would be a shame not being able to use this technology on other animal models. Thank you very much. And I leave the other person to talk. Romina.
Thank you, Marie-Christine. Thank you for that introduction. So I will go straight to the cheese because I think that Laurent did a very nice introduction of my role in MIRCen in the CEA and what I have been doing there for the past 12 years. I will talk now of the use of non-human primate models to study neurodegenerative diseases as a complement to what Marie-Christine has just shown of all the possibilities that we have in rodent models. I will not specifically touch upon the ethics, although this is something that we discussed with Marie-Christine before the seminar, because that will devour the 20 minutes of our talk. But if you have questions at the end, we will be very happy to discuss that. So the general strategy of MIRCen is to try and understand the mechanisms that underpin neurodegenerative diseases in the hope that we can develop and test new therapeutic strategies. The strategies can be disease-modifying strategies, regenerative strategies, or just treat symptoms. And the other main concern of MIRCen, because we're an imaging center, is also to develop and validate new imaging tools that are non-invasive in nature and that can be translated easily to the clinic. And this allows us to assess all types of dysfunctional neuronal dysfunction, cell death, and also therapeutic efficacy, and in some cases even target engagement according to how you create your ligands. But for all of these, obviously, we need pertinent animal models because many of these investigations cannot be carried out in patients. So when we look at how to generate a non-human primate model, we have two main classes of models, the phenotypic models and the genetic models. The phenotypic models we refer to as those that try to mimic parts of the disease or the whole of the disease, that's the aim, but not following the mechanism by which the disease occurs in humans. So, for example, for Huntington's disease, there's toxins that you can administrate. The 3NP toxin is an intramuscularly delivered toxin that specifically interferes with mitochondrial function in the striatum, and that can create lesions in the choroid and putamen, and that give a phenotype that resembles Huntington's disease. Otherwise, you can also use other toxins like quinolinic acid or ibotenic acid or cyanic acid. This also follows discoveries in patients of the kinurate pathway in Huntington's disease, but these are intracerebrally delivered toxins, so you target directly the areas that you want to lesion. And as you can see in the pictures on the top, you can study the phenotype, and we will see that. And you can also do histology to see where the lesions are in the brain and use MRI to monitor those lesions in vivo. For Parkinson's disease, you can use the benchmark standard molecule, which is MPTP. This toxin is injected most normally intramuscularly, but also intravenously. It was discovered in humans and then applied to rodent and primate models to generate a Parkinson phenotype. And as you can see from the pictures, you generate a degeneration of the dopaminergic cells in the substantia nigra that creates consequences in the striatum. As you can see on the PET images in the middle panel, this results in loss of dopamine. This is a unilateral model, so you have it on the dopamine on one side and not on the other. And then nowadays, we can also generate models by overexpressing proteins that are known to be the cause or affected, at least in Parkinson's disease. So it's a case of alpha-synuclein, but also in other diseases and like tauopathies or Huntington's disease. You will see a lot in the literature about injecting fibrils or by patient extracts. And in MIRCen, we do both of these things to generate models as well. The other type of models is the genetic models. As Marie-Christine just told you, it is possible to modify any organism nowadays, and with CRISPR technologies, the sky is the limit. This is not done much in France yet or in Europe at all. It's mostly the US and Asia that are leaders in genetic modification of non-human primates and generating transgenic primates. So what we do most normally is overexpress the proteins of interest or the gene that is of interest with a viral vector. We can use lentiviral vectors, we can use adeno-associated vectors, and we have generated in this way models of Huntington's disease by overexpressing mutant huntingtin, LRRK2 on alpha-synuclein, or the dementias, so using tau constructs or APP for A beta. And the advantage is that you control where this protein is expressed by targeting different regions stereotactically and injecting directly in the brain. The disadvantage is that you cannot create widespread expression, for example, in large cortical areas, because it's impossible to target the primate brain in that way. So how do we study these models once you have them? The first thing that you can look at is behavior, which is translational to the clinic as well. And for the Parkinson's and Huntington's disease models, for example, we perform quantitative assessments of locomotor activity. We use software that is developed in the Netherlands by Nautilus, but there's many ways of looking at locomotor activity. We tend to film animals and look at their spontaneous locomotor activity, or induce. You can also give a drug just before you film the animal. And you can, as you see in the pictures, you can detect the contour of the animal, follow its activity over time. And this generates unbiased, non-user dependent quantitative data. And as an example, I have put here a bar chart where you can see the baseline locomotor activity of an animal before it was lesioned with MPTP, then in green, the time when the animal was stably Parkinsonian, and then in orange, the effect of a therapeutic strategy on locomotor activity showing recovery over time. As I said before, this is not the only way of studying activity in primates. You can use collars, you can use activometric measures, etc. So there's many other ways of measuring activity, but this is something quite standard to do in primate behavior. The next thing you can do is wonder what about the quality of the movement and not just the quantity of the movement. So in particular for Huntington's and Parkinson's disease, patients present dyskinesias, abnormal and involuntary movements. And you can see the same things in primate models. For that, we also use another software developed by Nautilus, which is called the Observer. This allows you to program a sort of encoding. As you can see on the right-hand side of the screen, you have a sort of log, a time log. You can decide which are the parts of the body of the animal that you will score and what type of movements you're expecting to see. So, for example, I don't know, licking behavior or an abnormal movement of the arm or of the foot or this or that. So you program this, and then every time you see an event as you're looking at the video, you just type that you've seen that event, you create this log, and from that log you can create a sort of graphical visualization. If we zoom in, what that allows you to see is that each vertical line, each of the small vertical lines, is an event that you have seen and you mark it as an abnormal movement, and each horizontal line is a part of the body and the type of movement that you have seen. This is not user independent, of course, just like in the clinic when you're rating patients, it's very dependent on the raters. But it allows you to extract quantitative data from that. And for example, you can use a drug and see when the drug is metabolized, when it kicks into the system, when it starts reducing dyskinesias, and also if, for example, the drug has a particular effect on one type of dyskinesia and not the other. So this can be a neat tool to use in that respect. Next, you can look at fine motor movements of the hand, and here I just wanted to draw the parallel in between the setups that you can find in the clinic and what you can do with monkeys. So it's not extremely different. The main difference is that monkeys like to retrieve pellets instead of just playing with pegs, but this is just to give them a little treat and a reward for accepting to collaborate with us in these experiments. And you're just looking at fine movement of the hand and latency and precision. And then the most widespread study nowadays, or tool to monitor cognition nowadays, is the use of touch screens. So there is at least one commercially available touchscreen that you can buy for monkeys, which is the CanTab system, and was developed for humans and then adapted to monkeys. However, most laboratories develop their own screens with their own software. It's mainly because it's expensive and also because it's a little rigid in terms of programming new tests in. We also have 3 different ways of testing animals. Mostly you recommend to isolate animals in a noise controlled space so that the animal is focused on the task. But we have done extensive testing of animals in their home environment, which means with other primates around them, and also in social groups. I will not touch upon that today in the presentation, but if you are curious about this, please don't hesitate to contact me by email or by chat at the end. So I hope this works. What we do is train the animals to touch screens and then train them to perform particular tasks. We have 30 to 40 tests in the battery right now, and this is a very dynamic process because tests can be programmed in as you start new collaborations, as you talk to neurologists and clinicians that are interested to see something in monkeys and they ask us to program something in. So there is no limit to what you can do. There might be a limit of the number of tests an animals can perform over time. If the batteries of tests and the combination of tests are very complex, this can be difficult for primates, but up to 5 or 6 tests we have done without problem, and it also depends on the complexity of each test per se. So I would like to now spend a little bit of time telling you how these tools can be used to characterize a model of neurodegenerative disease in primates. And I have chosen the model of tauopathy. This is not published yet, but hopefully will be published this year. So we used AAV vectors to overexpress 2 tau constructs, mutant tau and wild-type human tau, in the hippocampus of adult monkeys. We followed them up for 12 months, and we wanted to understand the consequences of the aggregation hyperphosphorylation of tau in the hippocampus on the function on the function of the animals. So of course we used cognitive testing on touch screens, and we were able to evidence difficulties in deficits in a delayed non-matching to sample test to look at different delays over time so that you have a contribution of short-term and long-term memory. And the animals were tested at 4, 8, and 12 months post-injection, and a great advantage of the preclinical setup is that they were also tested at baseline, of course, and you could see that their deficits starting at 8 and 12 months, so blue and orange, in the mutant and the wild-type groups. This group is a benchmark lesion group, and the PBS are our controls. What's interesting as well when you're doing cognition is not only test whether the animal remembers a rule that they were taught before you started inducing a pathology, but also understanding whether they can learn a new rule and how difficult it is. So at 4 months post-injection, when we knew that there was some pathology starting, we taught them a new test de novo that they had never seen before. And as you can see here, the mutant group needed a lot more sessions of training to reach the same scores of success than the intact and intact group of animals or the PBS group of animals or even the wild type injected animals. And this is our benchmark lesion control. Then because they had learned this test at 4 months, we tested it again at 8 and at 12 months. And there again, the performance in terms of success score of the mutant group was lesser. And this test looks at spatial memory. I didn't put the data in here, but we also delivered, and it's very important to do so, control tests, so tests that should not be affected to disease. We delivered one test that was non-hippocampal dependent. And it is important because you need to know that this is not an effect of apathy or depression or motor or any other confounding element that could bias your interpretation of what is really happening in your cognitive test. And then we also delivered a specific mnemonic task, so like to look at memory, but we overtrained the primates at baseline so that the information was stocked somewhere else in the cortex and not in the hippocampus anymore. So if you don't need the mediation of the hippocampus to retrieve a significant part of the operation of that task, the task is not affected. And those are the results that we found. So it's nice to combine measures of both retention and rule learning, and also to have specific and non-specific tests combined in your battery. This is why I was talking about teaching animals about 4 or 5 tests every time you want to test even one single aspect of cognition. The other thing that we used to characterize the model was PET imaging, and here you can see to the left images of FDG, so this is glucose metabolism in the brain, and on the right you can see DPA714, which is a tracer for neuroinflammation, a TSPO marker. In the top row you see healthy controls, in the middle row the animals injected with wild type tau, and in the bottom row the animals injected with the mutant tau. And as you can see, there seems to be a gradient of decrease in glucose consumption when you go from healthy to wild type to mutated tau. And it's interesting also to see that the decrease is global. It's not just concerning the hippocampus and entorhinal cortex, but it concerns most of the cortex and not so much the basal ganglia. And this is paralleled with presence of neuroinflammation, which is mostly negative in healthy controls. It has an intermediate phenotype in the animals injected with a human wild type tau, and it really brightens up. As you can see in the image, it lights up when you inject the mutant construct. So both of these indications of in vivo functional follow-up, the behavior and the PET, go hand in hand together, showing that we have created some dysfunction. Now we obviously needed to see the pathology to understand what this dysfunction was due to. So we used an 88, which is a marker of phosphorylated tau, to look at the hippocampi of this animal at different anterior-posterior levels. Here I'm showing you a representative example, and you can see that the area is really darkly stained with 88. And we looked at other markers, PES422, 8100, MC1, several markers of conformational change and phosphorylation of the tau protein, and all of those were positive. We also looked at markers of neurons to see if there was a lesion, and there is no obvious loss in NeuN, and there is some inflammation when you look at IBA1 for microglia or GFAP, but not striking a neuroinflammation in the sense that we're looking for specific signatures of a lesion that could be non-specific. For example, due to the viral vector itself. If you have an area around the injection site where you have clearance of NeuN and a lot of inflammation, is most likely due to the toxicity either of your virus or the copy cell number that you have around the injection site that could create a deficit because you're creating a little hole in the brain, but would not be mimicking a pathology. So it's important to say that in this case, in this model, it was not the case that we had an unspecific lesion, and this was more due to the accumulation of phosphorylated tau. The other example that I would like to show you here is the use. So that was an example of a genetic model. Now I would like to give you an example of a phenotypic toxic model. So using the MPTP model in non-human primates to generate a Parkinson's disease phenotype, and then testing a strategy of gene therapy to restore and replenish dopamine in the striatum. This was performed by intraputaminal delivery of a lentiviral vector that you can see here that overexpresses TH, ADC, and CH1. These are 3 key factors in the production of dopamine in the brain. And the strategy deployed by Oxford Biomedica, the the pharma partner that contacted us many years ago to test this strategy in monkeys is to go from the physiological dopamine synthesis to this triple enzyme approach to be able to release dopamine in the depleted striatum of a Parkinsonian patient. So what we did at the time was look at locomotor activity with the etovision software as I showed you before, and here you have the normal activity of animals at the beginning. The MPTP activity severely decreased. That's the point where you inject your viral vector, and in blue you see the animals that have been treated with a prosavin gene therapy approach, and in red and gray here you see either animals that have been injected with a LAG-Z virus or animals that have not been injected at all. They're just remaining MPTP. So you can see both of the model is stable over time. These are 12 months follow-up, and also that there is a significant recovery in locomotor activity. Then we also look at the appearance of abnormal involuntary movements, dyskinesias, and here we compared only the curve in blue here, the animals treated with prosavin, to animals that had received the MPTP only and not the LAG-Z. And if you give oral dopamine, normally after a certain period of time and doses, you provoke abnormal involuntary movements, both in human patients and in primates. And we can see that the fact of treating these animals with the lentiviral vector preventing the dyskinesias from appearing. The final confirmation from this study was looking at the ability to synthesize dopamine in situ locally in the brain. So before euthanasia, the animals went through microdialysis experiments to measure the extracellular dopamine. As you can see, it's not fully recovered to normal standards, but it's significantly increased compared to the MPTP only and MPTP plus control viral vector injected. All of this data was published in Science Translational Medicine in 2009, and it gave rise to a clinical trial. The first results of the clinical trial came up a couple of years later, showing, as in all phase 1, phase 2 trials, that the vector was safe. They tested 3 doses of the vector in a small group of patients, and recently in 2018 we had a longer term follow-up. So the vector is still proven to be safe, and the efficacious dose was the highest dose used. In view of these experiments, in these experiments in primates, but also in the results of the clinical trial, Oxford Biomedica decided to go back to the bench and try to design a viral vector that gave rise to higher production of dopamine, because now that we know it's safe and that the highest dose is the one efficacious in humans, that made sense. And when they had it, they came back to us to ask us to test this in MPTP primates. And again, we looked at locomotor activity. We used clinical scores, which are scales that you use in the clinic and in the preclinic just to observe your animal. And we used the etovision locomotor activity. And here you can see that these are the control animals that have a very high Parkinsonian score, and these are the treated animals that have an almost normal score by 6 months post-injection. And you can see the same results when you look at locomotor activity at 3 or 6 months post-injection. So they have a severe reduction in locomotor activity when you render them MPTP, and except for the control animals, the other animals recover. And in pink you have the highest dose of the new vector, XB102, which is the most efficacious one. We also performed PET imaging in these animals, and we looked at this time at ADC activity. This also gives you an idea of the production of dopamine, and it has the advantage that one of the transgenes in the viral vector that we injected is the ADC. So the dual purpose of using this tracer is that you can see if the dopamine is restored in the striatum, but also have a direct visualization of the expression of your transgene in the clitting, which for safety and efficacy studies is very useful. So you see in the top row the normal expression of the ADC in the primate brain, the consequences of the MPTP lesion depleting the brain from ADC and dopamine, as we know, and then the consequences of injecting your viral vector, and this is at 6 months post-injection, and you have a restoration of that activity in the putamen. And of course, this brought Oxford Biomedica to the point of readiness for another clinical trial, which is planned to start this year. And I thought that this was a nice example of going from the bench to the bedside and then thinking about it and coming back again to the bench and to the bedside. And it's what Marie-Christine already said of this kind of virtuous circle of refining and iterating experiences and refining the treatments that you can propose to patients through animal experiments. And with that, I would like to leave some time for questions and remind everyone that research is a collective effort and that none of what I've shown you could have been done just in MIRCen. And thank you for your attention.
Thank you very much, Romina and Marie-Christine, for your nice presentation. We will now have time to address your questions. Please feel free to add posts to the question box. So the first question we have is rather for Marie-Christine. So many in the fields are skeptical about the requirement to study rodent behavioral activity as a must-have before moving to a CNS compound into the clinic. Will tools like CRISPR-Cas9 take away the skepticism by providing truly translational or more predictive models, or is more needed?
I suppose so. I mean, not sure I understand really well the question. I mean, you believe, I don't know if the person believed using other species or just using CRISPR to go, it won't change a lot. I mean, in the mouse, it won't change completely the field. I think in the mouse, we knew already how to do different models through ESL. We will use same kind of model. I'm not sure I'm answering really the question, but I mean, it won't change the way we do things. It's just a tool to, CRISPR is just a tool to go faster and to generate faster models. Then that won't change things. I think it will change because now it is possible to generate mutants in any species. I mean, for instance, we have already implemented rat, and CRISPR-Cas works really well in rat. And rat is definitely one model that is really interesting in behavior. I mean, it behaves much closer to human than mouse, then it gives really nice results. But for mouse directly, I don't think CRISPR will change a lot because we will just do the same in another way.
Thank you very much for your answer. Now we have a question for Romina. Very impressive results on the PD model. You are using tracers. Is the current toolbox of tracers complete, or are more or better needed? What role will these tracers play in the clinic?
So no, there's a lot of room for improvement. I would argue, and I think Philippe will agree with this, that the tracers have been the same ones for a very long time. The current chase now for tracers is obviously tracers that bind aggregates and specific proteins, so not so much the tracers to look at FDG, so general metabolism or the status of the dopaminergic system. You have several tracers looking at the DAT transporter, looking at F-DOPA, looking at FMT. So I think we're covered in trying to see what the status is of the functioning system. The same for 5-HT, but there's lots of other receptors, muscarinic receptors, that are not explored. So there's a lot ongoing still on that. And the big investments nowadays from pharma mainly and from patient associations is trying to get a tracer, for example, for alpha-synuclein, or a better tracer than PAB for A-beta, a tracer for tau. So tracers that are specific for the proteins, because one of the biggest needs in the clinic is to be able to stratify your patients, which is a big struggle and which determines the results of a clinical trial. If you stratified your patients wrongly and you have what you believe is a homogeneous population of patients, but it's not, then you will be biasing your results and losing power and not even knowing about it, and you have the postmortem. We have to remember that in the clinic, many of these diseases are still confirmed diagnosis only with a postmortem. So useful tracers for stratification would be great, and also for early biomarkers of disease progression and trying to see both if you're diagnosed correctly, and B, if your therapy is having any kind of improvement and it's a disease modification therapeutic. So, for example, the strategies are looking at antibody therapy, so ASOs that want to reduce the load of protein accumulated in the brain would highly benefit from radioligands that are specific and that would show results that correlate with CSF or other measurements. So it's far from being over. There's a lot of research to be done.
Thank you. Now we have another question for Marie-Christine. What is, in your opinion, the role of natural extracts in the improvement of cognitive functions? Is the green tea extract an appropriate option for improvements of cognitive functions for other diseases, not only associated to DKK1A?
Well, I mean, all the diseases where DYRK1A is overexpressed it would be interesting to treat them with green leaf. Then I am not specialist of behavior, and I'm not sure there are many other diseases in which DYRK1A is overexpressed, because DYRK1A is on the human chromosome 21. But I thought it was just a good example to show you that it's possible to find things that ameliorate the life of people, and mouse in this case was really a good model to test the efficiency of this treatment. Then, if there are other diseases in which DYRK1A is only specifically overexpressed, it would be a good thing to try it on the top of that. But I mean, yeah.
Thank you. We have now come to the end of this webinar. I would like to thank you all for attending today's webinar, with a very special thanks to our lecturers for their excellent presentations. Should you have any further questions to one of the speakers, I welcome you to submit those to us by email, and we will get back to you as soon as possible. The recording of this webinar will be made available to anyone interested on our website within 2 weeks. Our next webinar is scheduled on May 6th and will be addressing high-field MRI in neurodegenerative diseases. We hope to see you all again then. Thank you and have a nice day.